Hello everyone, I'm Shubhada Kulkarni. Today we will discuss about the history of microbiology. Yes, wait a minute. Yes, I think now it is visible to all of you. So, history of microbiology, that is a spontaneous generation doctrine. Now, how this history began, how these microorganisms are arised. So, let's see one by one. Okay, so if we are talking about spontaneous generation doctrine, so in that there are certain beliefs related to the how these organisms are arised, and at that time the superstition was that the organisms they are generated through directly through the organic infusions. Okay, so like that, the belief that life may arise from the non-living matter. If we put the meat broth inside the beaker, directly from that beaker, the mice, the rats, they are arise. Okay, so like that, the beliefs are there. So if we are talking about this particularly thing, so there is a controversy is going on in between the spontaneous generation and a spontaneous doctrine. Spontaneous generation means abiogenesis, and a spontaneous generation means biogenesis. So there is a debate is going on in between these two. So in the decade 384 to 322, so Aristotle, that is this scientist, he actually he was the first major proponent of the spontaneous generation doctrine. He believed that the worms, insects, or they can arose spontaneously from the soil or decaying organic matter. And so the first belief of this, that is the organisms or these life forms, they are created through the inorganic infusions or any organic non-living thing. So from that the new life are formed. So like that, the Aristotle at that time they proposed this theory. But after that, the in 1250, the Roger Bacon, he postulated that the disease is caused by invisible creatures. But unfortunately, he had no proof to prove the statement. And so for that, this work of Roger Bacon, or the postulate of Roger Bacon, is remain as it is. But after some time, the Girolamo Fracastiro and Anton van Plensis, both of them, they propose the same statement that disease is caused by invisible creatures. So all of we are aware regarding disease. So disease is an abnormal condition which is caused, which is abnormal condition which is caused by these type of organisms. Okay, and so at that time, both these two, Griolamo Fracastiro and Anton van Plensis, they are supporting the Roger Bacon that the disease is caused by invisible creature. This statement is correct, but they had no proof to prove their statement. And in our science and technology, there should be a proof is required. So after the work of these scientists, then the Anton van Leeuwenhoek came. Anton van Leeuwenhoek was the merchant. He was a scientist. He fond of collecting lenses. He collected the lenses and prepared the microscopes. His microscopes are spherical lenses and mounted between two metal plates. And if we are talking about these type of microscope, so the microscope of these Anton van Leeuwenhoek is it can give the magnification near about 50 times multiplication, or 50 times magnification was there, up to 300 times magnification was there. And Anton van Leeuwenhoek, he used to observe everything through these microscopes. Anton van Leeuwenhoek, he wrote more than 200 letters of observations of all these things from this microscope. And these observations, that is a 200 series of letters, they are published in the Royal Society of London. So the Anton van Leeuwenhoek was actually a great scientist who worked, he contributed tremendously in the microbiology. And one day, when he was observing through his microscope, he took the human teeth scraping, and in that, he found that there is a presence of tiny creatures, and these tiny creatures he called it as animalcules. And these animalcules nowadays we generally know as a protozoa. If we are talking about the first live cell, so the first live cell is invented by Anton van Leeuwenhoek, and so he is called as father of microbiology. If we are talking about only cell, so cell is invented by the Robert Hooke. There is a honeycomb like structure was there. But if we are talking about the live cell, so live cell is invented by Anton van Leeuwenhoek, and so he is a father of microbiology. So huge contribution in the field of microbiology by the scientist Anton van Leeuwenhoek. And again, he proposed one statement. He given one statement that these that is the on this particularly that is a spontaneous generation doctrine, the new life forms they are created through the eggs of these animals. This was a statement by the Anton van Leeuwenhoek. The new life form was created through the eggs, through generated through the eggs of animalcules. But the supporter of spontaneous generation doctrine, they are oppose this statement, and still the controversy is on and on and on. But after that, the next scientist come, that is a Francisco Reddy. Francisco Reddy, what he did, he actually, 1 minute, I will take the pen. So what he did, he actually took the containers, A container and B container like this, okay, and he put here meat broth inside it. He put the meat broth. Here also he put the meat broth. But this A flask is totally covered with the fine gauze. So this is a fine gauze. So like this, fine gauze was there. So by using this fine gauze, so this is our A flask, and this is B. Okay, so A flask and B flask. So this Francisco Reddy, he was supporter of spontaneous generation sorry, a spontaneous generation doctrine. What he did, he took the A container and B container, and in both of these there is a presence of meat broth, and he covered this first A flask by the fine gauze. Okay, so the porosity of this gauze is very, very small, and this B container remain open. After the incubation time, means after 24 hours, this A broth remain clear. It remain clear. But this flask B, it become cloudy and cloudy and cloudy and cloudy. So like this. So maggots are appear inside it. And so that's why the Francisco Reddy stated that the new life form is created through the eggs of animalcules. So like this, the statement was proved by Francisco Reddy. So he was supporter of a spontaneous generation doctrine. Then the next scientist came, that is John Needham. So John Needham is a supporter of spontaneous generation doctrine. So what he did? So he take the meat broth again. So I will take this pen, and he took the conical flask. So these are the conical flasks. He took this conical flask, like this, and he took the meat broth. He took the meat broth here. So meat broth is added into this. Here also the meat broth is added. So here the meat broth is there. Here also the meat broth is there. He boiled this for some time. Okay. He boiled it for some time. And after the boiling, he sealed this A flask by using cork. So like this, the cork is there. So he sealed it by using cork, and this remain open. Now when this sealed after incubation time, that is 24 hours, this broth become cloudy, and this broth also become cloudy. There is a maggots are appear in both these flasks. So John Needham stated that when we are sealing this flask by using cork, still the maggots are appeared. So he was supporter of spontaneous generation doctrine, that the all the life forms, they are created through the directly through the organic infusion. Okay. After John Needham's work, the supporter of spontaneous generation doctrine, they are working and working, and they are giving some certain statements. But after that, the next scientist came. The name of scientist is Lazaro Spallanzani. So he repeated the experiment of John Needham, but instead of heating the broth for a small time period, he heated the flask, that is which contain the meat broth, for long time period. And after that, he sealed the mouth. He sealed that particularly flask, but before sealing, he heated the mouth of the flask. And when the mouth of the flask is heated and then it is sealed, so at that time the heated air is enter inside the flask, which is not there in the John Needham's experiment. So if we are talking about John Needham, he directly sealed it, so impure air is enter inside the flask, and so that's why the maggots are appear inside the flask. But here Lazaro Spallanzani, he heated the mouth of the flask, so the heated air is enter into the flask. The heated air is a optically empty air which enter inside the flask, and so that's why the broth remain clear after incubation. But the control flask which is not sealed, which is so it become very cloudy. Okay, so like that, the statement of this sorry, the Lazarus Spallanzani, he proposed, or he supporter of a spontaneous generation doctrine, and he gave one statement that the air is the main source of contamination. So this was the very important statement given by Lazarus Spallanzani. Sorry for disturbance. So let's start again. So Lazarus Spallanzani stated that the air is the main source of contamination. So after that, the next scientist came. The name of scientist is Suez and Schwann. Suez and Schwann, what he did? So they performed the experiments that Schwann treated the air by passing through the heated coil. So I will show only one, so you can understood what I am telling. So I will take the pen. Yes. So this is the meat broth. It is present inside the flask. So this is the flask. And here, so yes. So like this, the heating coil is there. This experiment is of Schwann, and this is the other experiment of Suez. So the air is passing in the flask through the concentrated acid or alkali. So the air is passing through the concentrated acid or alkali. So this was experiment of Suez. So in these both flasks, that is flask A and flask B, there is, so in the flask A and B, there is a meat broth was there, and this air is passing in this flask through the heated coil. This experiment is of Schwann, and in this flask, the air is entered through the concentrated acid and alkali. So in both these cases, the air is treated, and so that's why after incubation, these both flasks remain clear. There is no maggots appear after incubation. So Suez and Schwann proved that the air is the main source of contamination. And if we are talking about the supporter of spontaneous generation doctrine, they opposes this experiment. Why they opposes this experiment? By stating that you are killing the vital force which is present inside the air. Okay, so like that, the proponents of the spontaneous generation doctrine, they are opposing this experiment of Suez and Schwann. Yes, sorry again for disturbance. So then the next scientist came, Schroeder and Von Dusch. So Schroeder, now just I stated you, the Suez and Schwann, they treated the air, and after they treating the air, there is no maggots appear after the incubation time. Obviously, the broth is boiled. After the Lazaro Spallanzani's work, the total broth they are utilized as a boiled broth. The broth, that is a meat broth, is heated for longer period of time. So this Suez and Chuan also took the properly boiled broth, and they both of them they treated the air, and there is no maggots there were no maggots appeared, and so that's why they proved that the air is the main source of contamination. But the proponents of the spontaneous generation doctrine, they are not accepting this, and they rejected this experiment by stating that you are killing the vital force which is present inside the air. And so that's why to overcome this objection, Schroeder and Von Dusch, what they did, they just take the boiled meat broth and he put the cotton plug toward to the mouth of the flask. So due to the cotton plug, the air can easily pass inside the flask without any treatment. Only the dust particles they get adhere to in the pores of the cotton. And so that's why till now we are using cotton plug in our microbiology lab. Okay, so if we are talking about this, so the Schroeder and Von Dusch, they proved that the not air actually, but the dust particles are the main source of contamination. And so like that, the controversy is going on and on and on and on. So after the work of Schroeder and Von Dusch, they invented the cotton plug. The next scientist come, the great scientist Louis Pasteur, and he performed the experiment. He prepared the swan neck flask for it, and he put the meat broth inside it, which is properly boiled. And after incubation time, the this neck is totally open neck. Okay, neck of the flask is open. There is no any cotton or etc. is added here. But still, after some time, after incubation time, there is no maggots appear inside the flask. And why? Because when the air is enter into the swan neck, so at that time, these dust particles they get adhere in the bending of the neck or bend of the neck, and so that's why the pure air is enter inside the flask, and so this particularly flask remain clear, broth remain clear. So he proved that that is Louis Pasteur proved that the that is spontaneous generation doctrine is not actually correct statement, as the new life it is created through the eggs of animalcule. The statement of Anton van Leeuwenhoek is correct, and so that's why biogenesis is a correct way of development of the organism. And after that, the final touch is given by John Tyndall. He prepared one box-like structure, and this box is internally coated with the glycerin. There is only one hole is there, that is a light, it can there may be a light beam can be passed through it. And when we keep our cultures, open cultures inside the test tubes or petri plates, inside that box, so at that time the broth or that media remain clear, free from the organism. There is no maggots are developed. Why? Because through that beam of light, that through that small orifice, the air is entering inside the box, but there is a presence of glycerin coating. On that, the dust particles get adhere, and the optically empty air is present inside the flask. And so that's why this optically empty air, it means the air which is free from the dust particles is present inside the flask, and so due to that there is no contamination occur and the broth remains safe. So here the our controversy of spontaneous generation doctrine has had a full stop, that the microorganisms or any new life form is generated through the eggs of animalcules, through the different reproduction processes. And so we can say like that the biogenesis is a correct way, that is a spontaneous generation doctrine is correct. Okay, so if for further details or further more videos I will upload along with the gate preparations. Okay, thank you.